Here, receptor protein S2, and this is the human membrane. Now, so in last couple of years, there is a revolution in structural biology, which is unprecedented. And this is actually enormously helping the drug discovery for this kind of work. So here, this is the what you are looking at, the crystal structure, of the SARS-CoV-2 protein and the AC2 of human cell. And this part will join the receptor. So here, this is the expanded version. You can see, so this is the SARS-CoV-2 protein, and this is the ACE2 human receptor. So now another strategy to block this is to flood the system with soluble receptor S2. So if you add soluble receptor S2, then S protein will be tighter out by the soluble receptor S2. As a result, the S protein will not get chance to interact with the cellular S2 protein. Thereby, the viral entry will be inhibited. So this soluble S2 receptor is in human clinical trial now. And also, by taking advantage of the artificial intelligence and machine learning, scientists can now mimic or even predict the structural prediction which will fit this place. So any molecule you can imagine that fit this place could be antiviral that can block the S protein S2 interaction, thereby inhibits viral entry into the cell. So bunch of compound has been discovered by this strategy through artificial intelligence, and many of them passed the preclinical evaluation and now in the human clinical trial. So you must appreciate now how this structural data is bringing a revolution in the drug discovery.
Excuse me, sir. Does remdesivir fit into this group?
Yeah, I'll come to that.
Okay.
I'll come to that. I have a slide for remdesivir. So if you remember that I told you that the virus has a seizure, that viral protease, virus, that 3CL pro. So if you can target the seizure, if the virus cannot cut the polyprotein and cannot make the small protein, so the virus cannot survive. So let's target the viral protease. So this is the structure. Gray area is the atomic structure of the 3CL pro, and this is a compound that's called ketoamide that can efficiently bind. All are made by structural biology, predicted by structural biology, and then actually validated. So this compound can block the 3CL pro, can bind the 3CL pro, and then inactivate the seizures. As a result, the seizure cannot chop the polyprotein and make those small active protein, thereby inhibits virus. Okay, here comes the remdesivir. So remdesivir is an inhibitor of RNA-dependent RNA polymerase. So this is the structure of the RNA-dependent RNA polymerase. This is the structure, and here you can see this is remdesivir. That how remdesivir fits into the structure of RNA-dependent RNA polymerase. So it is actually a nucleoside analog. So it is. So the RNA-dependent RNA polymerase think that this is a nucleoside, so it will bind and try to incorporate into the DNA, but it cannot, because it is a nucleoside analog, not a true nucleoside. So thereby, so if it cannot incorporate, so it blocks the site, thereby the chain extension is not possible. So that's how remdesivir works. So this is also currently showing very good result in human clinical trial. And not only remdesivir, one of its derivative called favipiravir. So this is also giving promising result in a recent clinical trial conducted by Harvard Medical School. Now I will try to show you, this is a really complex, I must admit. But this is the state-of-the-art drug discovery where it is pushing now. It is going now, the frontiers of drug discovery. So the virus, how, oops. So the virus will use its own protein to disrupt the communication between the cellular protein. So you imagine under normal condition in our body, in our cell, thousands of protein of human bodies or cell is communicating between each other in order to keep the cell efficient and healthy. So thousands of proteins are communicating with each other. So this is normal. That's how we survive. That's how we are healthy, because the normal communication happens between protein to protein in our cell. When virus enters the cell, virus uses its own protein, viral protein, and then tell go and disrupt the communication. So here I tried the color coding. So this is the virus entering the human cell. This is the normal cell, normal communication. This is the virus entering the human cell. So colored ball are viral protein, ash colored ball are cellular protein. And then you will see the square. It is the cross point of communication between viral and cellular protein. So viral and cellular protein cross point of communication is this square. And any future antiviral that will be effective, that should be able to break this cross point. So any compound that could efficiently break the cross point between the viral protein and the cellular protein could be a potential antiviral of the future. Excuse me. Now, I'm sorry, this is a little bit more complicated side. So now, you must have heard that there is drug repurposing. So this is a big deal now, because when pandemic happened, so there is not that much time to do all big long clinical trial, safety trial, all these things. So why drug repurposing could be efficient? Because if we could find an efficient antiviral from the already existing drug for other disease, but that drug also works for viral disease, that could be very effective, because already the drug is in the market, has passed the clinical trial, and safety, there will be no not that much safety issue. So repurposed drug could be very efficient, but how we will know? Even if you could predict that this drug could be anti-SARS drug or could be anti-coronavirus drug, how we could predict that? So in order to understand that, you have to understand the enormous complexity of the gene expression system. So now there is a site called Gene Expression Omnibus, or GEO site. So GEO site, people who are working on gene expression or high throughput gene expression, they are just dumping their data on that site. So all of the drug that is being used in the market, it's very likely that some people have done some work on change in gene expression in response to that drug and deposited that data into the gene expression site. So the people are going to that gene expression site, and by again using artificial intelligence, they are building this network that what kind of genes is being expressed by those known drug from the site. This is entirely data mining. So, and now they build this network. So this is again, this ash colored ball is the point of contact between the cellular protein and the viral protein. So the artificial intelligence build this network, and the colored ball is the point of contact between the cellular protein. So for normal function, normal physiological function. And the ash color ball is the point of contact between the cellular protein and the viral protein. And now they will compare this with the gene expression omnibus data that is they could retrieve in response to this drug, and then plot this network. And from this network, they can predict that which drug could actually disrupt this contact point. And then they are this is called the hits. So they are the hits from the artificial intelligence analysis, and then followed by real test whether this compound inhibits this virus. And many of the compounds that people have predicted actually working against this virus. So many of them are in the human clinical trial from this analysis now, as we speak. So is there any question up to this? So I don't know whether you guys are getting it. So I just want to know some of your feedback. Is there any question?
I think there are, because someone was raising hand. Tanmoy Ghosh was raising hand.
Yeah, I will entertain question. Okay, let's have some question.
Tanmoy, will you like to ask sir something? Tanmoy, unmute yourself. Can you hear me, Tanmoy? Sir, I think the questions will be answered at the end. You can.
At the end? Okay.
Yes, madam. I am hearing you.
Oh, want to ask anything? I
No,
think sir
madam.
has explained very nicely. So
Very nicely.
yes, sir,
So,
you please carry on, sir.
no, but if you have any question, feel free to ask through your professor. Okay.
Okay.
So okay, now let's move to the immune response of this virus. So you have to understand that there is a previous version of this virus called SARS-CoV, and a lot of immune response studies has been done, and there is a very high resemblance of immune response. So this is what you are looking at. This slide is a classical immune response against the virus. How our cell react to the virus. So you have to understand that virus itself do not cause much damage. Virus is not killing us. But who is killing us? It is our own cell. How our own cell behave in response to the virus. Sometimes that response goes ballistic and causes hyperinflammation. That overactive innate immune response, that is the cause of the death, not the virus itself. So now let's discuss the classical immune response pathway of this virus. So when virus enters the cell, I told you that many proteins have ion channel activity. So these protein can turn on the NLRP3 caspase 1 mediated inflammation, and form inflammasome. This inflammasome converts pro-IL-1 beta to IL-1 beta, and then IL-1 beta is highly inflammatory, so it's coming out from the cell. Another pathway is when virus release its genetic material, that's first the single-stranded RNA. That single-stranded RNA itself causes through TLR7 mid-88 pathway and through NF-kappa B pathway can translocate this activates NF-kappa B, translocate to the nucleus, and then causes activation of many pro-inflammatory cytokines, including IL-6 and TNF-alpha. You know that IL-6 is one of the culprit for the ARDS that caused by this virus. Again, another pathway is the double-stranded RNA dependent pathway. So when virus enters the cell, it releases the single-stranded RNA. So when single-stranded RNA forms double-stranded RNA, and when double-stranded RNA form, this double-stranded RNA activates the RIGI, MDA5 and MAVS mediated pathway that eventually turn on the IRF pathway, interferon response factors, and that translocate to the nucleus and then binds to the ISRE, interferon stimulated response element that harbours into the main interferon upstream region gene and then activates interferon. So lots of interferon will be made. This interferon itself is antiviral, and that interferon will go and bind the interferon receptor of the cell itself, and that binding itself turn on a signal transduction pathway, JAK-STAT mediated pathway, and that will again turn on more more inflammatory molecules like CXCL10, RNase-L, and why it is happening? Because all of them is actually working against the virus, want to kill the virus. But these are lethal molecules. If this goes uncontrolled, then this guy can kill not only the virus, but also the host cell. This is the reason why people are dying. The overproduction of inflammatory molecules. Okay, when virus coming out of the cell, this is the new virus, newly synthesized virus. When it is coming out of the cell, then immediately in the system, it will be challenged by the macrophage. This is another cell of our innate immune system. So this virus will be challenged by macrophage, and you know the macrophage is the antigen presenting cells. So macrophage will engulf the virus, and then it will display the viral antigen that will recruit the naive T cell. So naive T cell will be recognized by the viral antigen, and that will cause the transformation of naive T cell to CD4 plus T cell, CD8 plus T cell. So this will, so cytokine causes more cytokine. So this will go like a vicious cycle. So if we fail to control this, it will cause hyperinflammation and will cause death. But this is also an important part of the innate immune system. So cytokine production, this B cell, you know what B cell does? B cell make antibody. So antibody will be made, and then this antibody will block the virus. So everything is happening for good, but it is for when people are dying, it is happening too much, which should not. So if you can control inflammation by using proper anti-inflammatory drug, you know, then that is also another clinical strategy to reduce the death. Here you are looking at a severe lung pathology. What's happening in ARDS that caused by this virus? But this is the main reason why people are dying by this virus infection. Not the virus itself. It is by the hyperinflammation in the lung. Here you are looking at the infected patient lung, and here you are looking at the post-mortem sample of the infected patient. That this is a color contrasted electron microscopic image that you can see that how SARS-CoV get into the lung cell. This is a pictorial representation of the human lung. This is the alveoli. So when in normal lung supports, what's the function of the lung? Lung supports actually carbon dioxide oxygen exchange. But when infected lung, what happened due to the super inflammation, this space is filled with bronchoalveolar lavage fluid and full with this highly expressed this cytokine. This is called cytokine storm that is causing hyperinflammation and death. Why? Because it blocks the carbon dioxide oxygen exchange. This is the reason why the people need ventilator, and more long period that a patient is staying in the ventilator, less chance of survival. So this is the reason that cytokine storm is causing everything. So targeting this cytokine storm by either by soluble receptor or the antibody is now also a major initiative. Here I told you that repurposed drug. So already many drug, anti-inflammatory drug against other purpose like arthritis. Anti-arthritis drug is also working very good here because they are causing a down regulation of this cytokine storm. Uh, Now, I will tell little bit more intricate balance that is happening in our immune cell, and absence of that balance is causing the hyperinflammation. So that immune cell here can be grossly divided into two kinds. One is inflammatory cell, another is anti-inflammatory immune cell. So a large number of immune cell with varying status of inflammation is there, and these cells are plastic in nature. Means plasticity means they can convert from one type to another type. So the relative number of these diverse immune cell actually determine the resultant inflammation in infected patient's body. So why? So here you are looking at the dynamic equilibrium of the inflammatory immune cells and the anti-inflammatory immune cells. So inflammation, you have to understand, inflammation is very complex system. So it is a double-edged sword. So if it is too low, you will be susceptible to infection. If it is too high, then also it is lethal. So there is a nature is making a balance between these inflammatory and anti-inflammatory cells. And then absence of this homeostasis can cause cytokine storm. If this balance is defective, then those patients can experience cytokine storm. So this is the reason of hyperinflammation and death of majority of the SARS-CoV-2 infected patients who are severely ill. Lack of balance between anti-inflammatory and inflammatory cells. And the cells that could maintain the balance is called with trained immunity. Sure, those cells are showing trained immunity and may not have severe outcome. So now it's a very interesting observation that people that they are staying in relatively germ-free condition, that means so-called healthy condition in Western world. So this balance is very less. So this is the reason why in USA there are many deaths, in Italy, in Europe there are more deaths than India. So India now lot of infected patients are infection is happening, but still if you consider that the number of people dying is way less than other part of the world. And why? There is a reason. Because most of the people in our country do not live in a germ-free condition, and it is good that they do not live in a totally germ-free condition. That means the system is under trained immunity. So this balance is more. Some people, of course, will have lack of balance and will have severe phenotype. But this is the general scenario of a country where there is no germ-free condition. So let's talk. You must have heard about the BCG vaccination. So what is BCG vaccination? So the percentage of, and it is known that the people with BCG vaccination are less prone to this SARS-CoV-2 infection. And this you will see very clearly, the percentage of severely ill patient and the death from the total number of infected people varies widely between country to country. Portugal is very close to Spain. Spain very high death rate. Portugal less death rate. Spain, no one took BCG. BCG was not a norm, but Portugal people took BCG. Even in African countries, in Japan, in India, the countries that they have used BCG, their death rate is less. So in addition, so what BCG is? So in addition to the BCG is against tuberculosis, but in addition to its specific effect against tuberculosis, BCG vaccine has off-target beneficial effect. That means it can induce trained immunity. So a large number of beneficial mechanism of the beneficial off-target effect of BCG vaccine. That mechanism is also known, that is inducing metabolic and epigenetic changes. So there are lots of work has been done on this. So the result, now there is a result from the randomized control trial in Australia. So the result is published just few days before. I just saw the result. And there is 53% protection by BCG vaccine. So I would think that people who, BCG should be another defense in country like India. I mean, people should get BCG vaccine. And there are many, detailed work has been done on this BCG vaccine, that how BCG vaccination protects against experimental viral infection. It's an anti-tuberculosis vaccine, but it protects also against the experimental viral infection by inducing the trained immunity. So Cell, Host and Microbe, the very important journal, very highly prestigious journal, is published this work. And also BCG-induced protection, how it developed trained immunity. There are big detailed review also published. So there is lots of literature that how BCG vaccine could help. So again, so now slowly we will go to the vaccination part. That is the last part of my talk. So to get the natural immunity, how you could get natural immunity? By getting infected, by the natural infection. But that may come with a price to pay. That if you do not have trained immunity, then you may be seriously ill, and that could be devastating consequences. So that's the reason why we need vaccine. So vaccine is a process of inducing artificial immunity by causing the structural antigen, by using the structural antigen or a weak version of the virus, by exploiting the same adaptive immune response. So instead of virus, you inject the vaccine, and then it goes to the same pathway, inducing antibody. That means the viral antigen will be present by APC or macrophage. It will activate the T cell, CD4 plus T cell, CD8 T cell, produce cytokine, and then induce B cell, and then B cell make antibody. And not only B cell can make antibody and also can make memory B cell. That means that B cell will remain in the system. And if you encounter with the real virus, then memory B cell will immediately start making the antibody in response to the virus. This is the advantage of the vaccine. So you must have heard the name of vaccine from Oxford Jenner Institute. So please keep an eye when this session will end. Maybe I need one more entry.
Sir, I think we should discuss the vaccine portion in the next session. May I end here?
Yes, let's discuss vaccine portion in the next session. And that's the end. That's the end part. We are very close to the end now.
We are joining back for the end session.
Okay.
I'm just ending here.